Hello, welcome to the video recap from today's first live lesson for Year 9. This took place on January 25th, 2021, just for those of you keeping track. So, when someone first peered down a microscope at a slice of cork and drew the boxes they saw, little did they know that they'd seen the building blocks of every organism on the planet. All living things are made of cells. Cells can be either prokaryotic or eukaryotic. Eukaryotic cells are complex and include animal and plant cells. Eukaryotes are organisms that are made up of eukaryotic cells. So the different parts of a cell are called subcellular structures. Most animal cells have the following subcellular structures. To make sure you know them all, we're just going to go back through them. So if we start with the nucleus. So the nucleus contains genetic material that controls the activities of the cell. Cytoplasm is a jelly-like substance where most of the chemical reactions happen. It contains enzymes that control these chemical reactions. The cell membrane holds the cell together and controls what goes in and out. Mitochondria are where most of the reactions for aerobic respiration take place. Respiration transfers energy that the cells need to work. And ribosomes are where proteins are made in the cell. Now, plant cells usually have all the bits that animal cells have, plus a few extra things that animal cells don't have. So chloroplasts are where photosynthesis occurs and makes food for the plant. They contain a green substance called chlorophyll, which absorbs the light needed for photosynthesis. A rigid cell wall made of cellulose supports the cell and strengthens it. And a permanent vacuole contains cell sap, a weak solution of sugar and salts. Cells are the basic building blocks and they make up every living organism. As you know from the last biology topic, specialized cells carry out a particular function. These specialized cells form tissues which form organs, which form organ systems. So if we try and match up these keywords to the definitions, cells are the building blocks that make up living things. Tissues are groups of similar cells working together to carry out a function. Organs are groups of different tissues working together to carry out a function. And organ systems are groups of organs working together to carry out a function. Sometimes the word function will be written as a job. So if we look at this particular example, we have a specialized cell called a muscle cell, and several muscle cells work together to form a muscle tissue. And different tissues, muscle tissue and fat tissue, will work together to form an organ like the heart. And the heart is part of a system, so it works together with arteries, veins and capillaries to form the circulatory system. And then the circulatory system makes up part of the human body and works together with other organ systems to keep us alive. So a little challenge for you. Pause the video if you're watching until you have an answer. Put the following components in order of size from smallest to largest. So the answer is epithelial cells are smallest, then epithelial tissue, then lungs, and then the respiratory system. Chemical reactions, there are thousands of different ones going on inside living things. These reactions need to be carefully controlled to get the right amount of substances. You can usually make a reaction happen faster by raising the temperature. This would speed up the useful reactions, but also the unwanted ones, so not very good. There's also a limit to how far you can raise the temperature inside a living creature before its cells start to get damaged. So living things produce enzymes that act as biological catalysts. Enzymes reduce the need for high temperatures, and we only have enzymes to speed up the useful chemical reactions in the body. Enzymes are large proteins, and all proteins are made of chains of amino acids. These chains are folded into unique shapes, which enzymes need to do their job. Chemical reactions usually involve things either being split apart or joined together. Every enzyme has an active site with a unique shape that fits onto the substance involved in the reaction. Enzymes are really picky and they usually only catalyze one specific reaction. This is because for the enzyme to work, the substrate has to fit into its active site. If the substrate doesn't match the enzyme's active site, then the reaction won't be catalyzed. The diagram at the top shows the lock and key model of enzyme action. This is simpler than how enzymes actually work. In reality, the active site changes shape a little as the substrate binds to it to get a tighter fit, and this is called the induced fit model of enzyme action. So a few questions. How do enzymes affect the rate of reaction? Enzymes will increase the rate of reaction because they act as biological catalysts. What is the active site of an enzyme? The part of an enzyme the substrate fits into is called the active site. What is the lock and key model of enzyme action? The lock and key model is a simplified model of enzyme action. It states the reaction will only be catalyzed if the shape of the substrate exactly matches the shape of the enzyme active site. So the substrate fits directly into the active site. Changing the temperature changes the rate of an enzyme-catalyzed reaction. Like with any reaction, a higher temperature increases the rate at first. But if it gets too hot, some of the bonds holding the enzyme together break. This changes the shape of the enzyme active site, so the substrate won't fit anymore. The enzyme is said to be denatured. All enzymes have an optimum temperature that they work best at. So a few questions. What is an enzyme? An enzyme is a chemical that speeds up the rate of reaction without being used up itself. What do enzymes do in living organisms? Enzymes catalyze or speed up the chemical reactions in cells. So chemical reactions like respiration, protein synthesis, and photosynthesis. Enzymes also break down large molecules into smaller ones and build large molecules from smaller ones. And they can also change one molecule into another molecule. Biological washing detergents contain enzymes. Explain why clothes washed at 60 degrees Celsius will not be as clean as those washed at 40 degrees Celsius. 40 degrees Celsius is the optimum temperature for many enzymes to work. So they can break down proteins in stains. At 60 degrees Celsius, the enzymes are denatured and the active site changes shape and cannot catalyze the reactions. The pH also affects enzymes. If it's too high or too low, the pH interferes with the bonds holding the enzymes together. This changes the shape of the active site and denatures the enzyme. All enzymes have optimum pH that they work best at. This can be very different from one enzyme to another. Okay, a little recap of digestion. In the mouth, teeth are used to cut and grind food into smaller pieces. This is physical digestion. It allows food to pass through the digestive system more easily and increases the surface area of the food to speed up chemical digestion. Muscles in the stomach will also help to physically digest food by squeezing it. Muscles in other parts of the digestive system also squeeze food to keep it moving by peristalsis. Even small pieces of food cannot pass into the blood. Enzymes are produced in some parts of the digestive system and break down food into very small soluble molecules so they can be absorbed by the blood. This is chemical digestion. Most enzymes work inside cells, but the digestive enzymes work outside cells. They're produced by cells in glands and in the lining of the gut. The enzymes pass into the gut to mix with the food. Carbohydrates are digested into glucose and other sugars. Glucose is used for respiration and to make new carbohydrates. There are 3 groups of enzymes in digestion. The first is carbohydrates. Carbohydrates break down carbohydrates into simple sugars. Amylase is an example of a carbohydrate that breaks down starch. Proteases break down proteins into amino acids. The amino acids are built into new proteins inside the ribosome. Protein synthesis is also catalyzed by an enzyme. Lipases break down fats into fatty acids and glycerol. Fatty acids and glycerol are used for energy, to build cell membranes, and to make hormones. Some questions for you. Why is physical digestion important? Physical digestion breaks the food down into smaller pieces to allow it to be swallowed and increase the surface area to speed up the chemical digestion. How is chemical digestion different to physical digestion? So physical digestion breaks the food into smaller pieces. Chemical digestion uses enzymes to break the food down into different products. What type of molecule is broken down into amino acids? Proteins are broken down into amino acids. What are the products of lipid digestion? The products of lipid digestion are glycerol and fatty acids. In which organs are proteases produced? The stomach, pancreas, and small intestines. Bob is eating rice, which is a starchy food. Name the enzyme that will break down the starch during digestion. Which part of Bob's body produces this enzyme? So the enzyme is amylase, which is an example of a carbohydrate, and it is produced in the salivary glands, the pancreas, and the small intestine. This is where the live lesson for this week ends. If you have any questions about the topic we've recapped today, type them as a private comment on Google Classroom. You have been set one past paper question on a Google form, so go back to Google Classroom and complete that now. Until the next video, see you later.